بسم الله الرحمن الرحيم. السلام عليكم جميعا and welcome in the Arab Week for Science. This session will be in English unfortunately, وأنا أعتذر يعني مقدما للجميع عن تقديمه باللغة الإنجليزية يعني بسبب مني أنا، نقص مني أنا، صعوبة ترجمة كثير من المصطلحات بالنسبة للغة العربية. فالموضوع السيشن اسمه الجينومكس 101 كما يعني الموضوع أو التايتل يوضح إنه احنا نتكلم عن الجينومكس أو بالعربية، هذه كمان تعلمتها زيكم، المجين، علم الجيناتكس 101 كمبتدئين أو متوسطين في هذا المجال. What's new and what's the history and where are we going. فأنا اسمي ملاك عابد الثقفي، أنا طبيبة باحثة ومتخصصة بالجينات الجزيئية أو الجينات الطبية وعلم الأمراض. So when we start the overview of these lectures, we will really have three pillars. The first one is about looking at the history, because it's really important to know where we started and because this history will somehow has influenced where we're going in the future. We'll also shed light on some of the methods being used over the histories. And also we will look at different sequencing technologies, as well as we'll talk about the structural variation, different gene identification method. Of course, we cannot talk about genome without giving a lot of focus of what we call it Human Genome Project or مشروع الجينوم البشري. And we also will talk about the applications, because it's very important to understand. Some of you maybe just want to know what is genome, some of you may be in the medical field, some of you may be in the scientific field. And this is really touching each one of us, regardless where we are, as a researcher, as a healthcare provider, or even as an audience who wanted to know more how this will affect us. So this is in يعني outlines in general. History. Although the history of genome go actually beyond beyond 1950, but I like to start from here usually because this is very important time point in the modern history of genome. In 1953, when the famous paper at Nature published by Watson and Crick described the double helix of the chromosome. What is a chromosome? Chromosome is a structure where we have in our cells 23 pairs of it, 22 somatic في الخلايا الجسدية وزوج من 6 chromosome في الخلايا الجنسية. Chromosome have all of our genetics materials and it got saved in our nuclei في النواة في كل خلية بشرية أو خلية في أي كائن حي. Because of this very important discovery and how it's exactly structured in our cells, two scientists got awarded Nobel Prize in 1962 for their works. And then after that, let's go to another important timeline, the 70s, where Albert Knudsen, another famous discovery made, where he described that his double hit theory, not the tumor suppressor gene, where he actually studied a specific gene. It's called retinoblastoma. Retinoblastoma gene الذي يسبب سرطان نوع من سرطان العين. But what he studied, he discovered that هذا الجين need to hit to develop this kind of familial أو الأورام الوراثية للعين. The first hit, you born with it. You inherit it from your parents. And the second hit happened with life. So to develop this cancer, you need this 2 hit. And that's what they call them tumor suppressor gene concept. So this was important discovery for at least in the cancer biology to understand it. And so another important point where in 1975, Frederick Sangers, many of you know it, a famous British scientist, big institute in UK named after him. And he's only one of the 5 people, used to say for a long time, only 4 people got Nobel Prize twice. Now this year we had another person who got the Nobel Prize twice, Barry Sharpless. He got it twice, mashallah. soon we hear somebody from the Arab world. Of course, we always forget المرحوم Dr. Ahmed Zouel, Allah يرحمه. He was the only and the first Arab until today who got Nobel Prize in any scientific field. So Friedrich Sanger, what he did, he described a method how we can sequence or read these genes or genetics material inside the chromosome of human cells. Because of that, he was awarded a Nobel Prize in chemistry. Second one, the first one, he was related to the description of the insulin structures. So it's an important timeline because this technology is what we used later in the Human Genome Project. But before that, another important technology got discovered, Kerry Mellis, 1983, where he discovered what we call polymerase chain reaction, PCR. I think everybody knows what is PCR after Corona. I mean, even if you don't know anything, you know, لازم أروح أعمل فحص PCR. فيعني everybody know. Basically, it's a really fundamental molecular tool or testing way. We copy it from bacteria. So bacteria has this chain reaction. It used it to, it's long a fight with viruses over the time. So it has this tool to multiply copies for certain pieces of the genetics material. Mellis awarded again Nobel Prize for this technology in 1993. And this is again tell you, it's two examples how just by inventing a technology related to genome somehow you can get Nobel Prize. Another famous example, I did not put it here because I still wanted to see more adoption for it in the field. Last 2020, many of you know, two female scientists won Nobel Prize again for a third technology. We learned it from bacteria, CRISPR. Many of you know it. But we still need to see more applications. So I think it to be how we can really affect our life. After 1983, very important meetings happened in the US in 1988 to discuss what we wanted. We wanted to start and do the first human genome sequencing. What scientists wanted to do at that time, we want to know exactly where each base pair of this genetics material located in each chromosome in our body. So we want to know the order, where they are located exactly. This is two famous figures of this meeting, of this project, Francis Collins and name now gone for me. It could come back. They worked on it, and it's started actually in 1990, and it took them 13 years. It actually took a shorter time than they thought. Originally, they thought they would finish in 2005. So this is just a bit of history to tell you where we are and how it started, at least. A famous pictures of Crick and Watson and in front of their 3D module of the DNA structure. I will not go very deep. Many people said they took advantage of another female scientist who showed them the X-ray of it. All of them are great scientists, and unfortunately, she died before the announcement of their Nobel Prize. I think if she was alive, there will be three of them who won this Nobel Prize. But this is what they basically did. They just, as we said, showed us how the genes are organized, are packed inside this human cell or human nuclei, around 2 meters of chromosomes, subhanallah, condensed, and inside this human cell, which we cannot see by our Reunan and Majarrada. Many of them, you need a microscope. So imagine inside it all of this complicated structure. Imagine all the spaces of DNA that's located in the structures. Now we'll just go quickly fast forward. A few weeks ago, if you are watching or following Nobel Prize account and news, so this year the Nobel Prize again in physiology or medicine went to Professor Zafinto Papue. I hope I am pronouncing it right. He is a Swedish genetist or revolutionary genetist, if you will. He won it solo, which something we did not see for a quite long time. For somebody in medicine and physiology to win the prize alone for his discovery concerning the genome of extinct humans and human evolution. So what is Professor Papue did exactly? I will take you to another short history. So in 1856, There was a group of labor, Amal Hafr, in Germany, in a place called Ninader Valley, Wadi Ninader. They were doing just digging or something. I don't know what they were looking for. So they found few bones and stuff. They thought it's bones of a bear, a dub ولا شيء كان مدفون أدبهم. They put it, they sent it, went to then they discovered it's not actually a bear, they turned to human, and then they give it the name of Ninadortolus. It's actually referred to the first kind of the human that we know about. They lived around 400 years ago, and they extinct maybe 40 or 30 years ago. So what Professor Papio did, he was a postdoc in the late 80s and the 90s, and he got advantage to study the DNA of this creature. And what he did was amazing, because imagine having a sample that almost 40,000 years old. How are you going to get a DNA out of it? And we'll discuss on a later slide. It's extremely difficult work. It's not easy. Add to it doing the complete sequencing of it. Not only he only did the sequencing for this creature, he also discovered another species, or it's back to the old human. It's called Denisovian. It discovered from, they did a sequencing from a finger that they found it in Denisova in Siberia. So he completed of these two extinct human species and complete sequencing of them. And again, these two are extinct almost 40,000 years ago. They lived 400 years ago before that. Most of the, if you want to look how much of their DNA it's in our body, if you are more in the Western European, you will have more of the DNA of the Neanderthals, around 4% or so. If you are more in the East, Philippine, etc., Melanesian, you will have Denisova DNA more. At some point of history, these two, they got married separately and even together with the modern human who we know today, Homo sapiens. And that's why we still have some remnant of their DNA in our bodies. But that's the reason why he got it. He really developed techniques and a way to look back at this very old, difficult sample and to put a complete sequencing for them. Okay, what is genome? I like this definition in Francis Collins. He's actually one of my role models in genomics of all times. They call him in the US the gene hunter. He worked a lot his entire career in the old method by positioning and looking for location of the gene, famously the cystic fibrosis gene. And then he led the human genome, and he led the NIH for many, many years, I think until just last year or early this year, he stepped out. He gave three simple definition. Think about it as our shop manual with an incredibly detailed blueprint for every cell in our body. يعني دليل, subhanallah, Allah, he put it. It's very detailed where every base pair is located, and give us direction who we are for each cell. Or it's a history book with a narrative of the journey of our species through time. And this is, I think, the discoveries made by Papio and others showed us how back we can go back at least to 40,000 years ago. So it's our destiny was written in this, and we still keep part of it in our today DNA. It's also a transformative textbook of medicine with insight that will give healthcare provider new powers to treat, prevent, and cure diseases. We see it today, we witness it. We can diagnose diseases, we can treat some of them, and we can prevent some of them. We can also cure some of them, not everything we want. So this is my favorite philosophical definition, if you want. But there is more scientific definition. We'll go over it in the next slide. So one of the fun facts or facts people thought before we do the genome, human genome, that human can have up to 40, and some of them said up to 100,000 genes. We did not know what we're having. And we know they have chromosome, double helix, they're a gene, but we have no idea what exactly we have or the correct number. So after the Human Genome Project was completed, it showed that we actually have around maybe a 20,000, 25,000, and the real number, the last updated number I looked at, it's exactly maybe around 21,283 genes. That's what we know until today. So we're not really far off from fruit fly, from C. elegans, and a human here. And this is the rice. The rice has more genes than us. And the maize, the corn, that have more genes than us. So it's not about the number of genes. It's about what the function of this gene and what kind, how they can change, what protein they produce. And again, this is a visual mirror. So this is the entire genome. This is all the chromosome, had all the genetic material inside one cell nucleus. Zoom in. Each chromosome has different genes, which is a protein coding area, and then intergenic area, non-coding area. This is, again, if you want to look more, if you care about the chemical structure, this is just a small piece of the DNA, which can inside any of the chromosomes. It has what we call the 5' end and the 3' end to code for a specific protein. The exon, with the coding area, to actually get translated to what we call messenger RNA. And intron, which we thought for a long time, when you hear the term junk DNA, it's not junk, it has some functions. We still don't know exactly every single function. But when they get translated to mRNA, we know exon get translated, and it has this attachment of polytail A. And then what is really important, at least for in the view of many scientists, is the protein that get transcribed from mRNA. This is what make most of the function in human body. Some of the protein will be enzymes doing different things. Some of them will be hormones, etc. So this is really what they do, the function. So think about it. This is a book. This book now got printed to make other things, and this is the final product. I mean, in the factory, this will do exactly what will do the function. This is why you hear some technology called proteomic. Some would call RNA-omic or RNA-seq, and some look at DNA. So there is different level of function in the gene level. So what is the genome again? It's the complete genetic constitution of organism encoded in nucleic acid, everything we have. What is the gene? It's only a small piece that code for a specific protein or specific function. So it's gene is part of genome. That's why many people do it interchangeably, but when we say genetics, we really mean looking at one gene, not the entire genome. The Human Genome Project, the most important project we might have the last 100 years, at least in the genomic field and molecular biology field. We wanted to know where each of these 3 billion base pair inside our genome, we actually came with the correct number, and we wanted to know how they exactly organized inside the chromosome. This is a 4 famous chemical base, ATGC. As we said, we now know we have around 21,000 genes coding protein. We know we're very similar, but we have very different. So we are similar, almost 99.6%, but this a few changes that would make us different in different traits. And we know we have some, we're not very far from other species, like, for example, to chimpanzee, we are almost 99% similar to the genome. However, there is this different what makes us human. It took them 13 years, as we said. The sequencing involved more than 20 universities and research centers around the world. Countries included United States, United Kingdom, France, Germany, Japan, and China. Although it's only famous to be called a human genome, but however, on the side, they actually sequenced other organisms at the same time, including bacteria and E. coli, bakery yeast, fruit fly, nematode, and mouse was sequenced to put their genomic sequencing. This is an old newspaper advertisement where they wanted to recruit the volunteer to participate in this project. We don't know who are these people until now. They say it's around 12 people who participated. We don't know. Of course, no personal information was shared, and they're still anonymous until today. But what we know, unfortunately, majority of them were of Western populations. Okay. So did they really produce a perfect genome the first time around? This is why you heard sometimes the project completed in 2000, sometimes in 2003. In 2000, they announced the first draft, which they could able to sequence using at that time only 90% of the human genome. They mainly used Sanger technology at that time. So the draft, it has more than 150,000 area were unknown because they could not determine accurately what is it. They call it gaps, فراغات في الجينوم. So 90% draft was published first, and then 3 years later they could do better. So they said now they show the draft which has 90% completion and less than 400 gaps. But look at now down. Just actually almost 7 months ago, we completed the full draft of the human genome, thanks to the project called Telomere to Telomere. So this is just now, they think we completed the first truly complete human genome sequence. And this last piece, this last 8%, a number of genes and DNA in it, it's compared to the size of the entire chromosome. So it wasn't a small amount. It actually, the more you read, there are over more than 2 million variants and 68 plus genes that was completed. There was gaps. And telomere to telomere, we'll talk about it in the next few slides, but this project were possible. For those who don't know, telomere, this is this piece at the end of the chromosomes, very end piece. They said with, many research said with age, this is what causes longevity, or which age is decrease actually. And this is what gives them immortality for some cancer cells because it's elongated or somehow. But telomere to telomere was possibly done project because of the advances of the technology. We're going to discuss it later. It's an odyssey. This is the Science cover page for the first draft in 2000, the Nature cover page of the second draft in 2003, the Science again cover page for the telomere to telomere in March this year, 2022. Do we really did it 100%? It's not really 100%, but we're really getting closer. Technology still need more advancement. I think we now reach 99.8%, to be fair to them. After the first human genome, multiple other international projects was started. Famously, we'll talk about two of them: international heat map or heat map projects, and also the 1000 Genome Project. And then we can start to see what we call a genome-wide association. So do a lot of genotype. Genotype is not meet. You look for a specific point. You don't really read the entire sequencing, specific point trying to make a correlation. So the international HabMap projects, they did more sequencing. Remember, the human genome was only on maybe 12 people. Now they look at 27 individuals from the 4 population. Now expanded a little bit from Utah to represent people from Northern and Western Europe. Trio, they took 30 families, so usually the parents and a sibling. And they also took Han Chinese and Japanese and some African candidate from Nigeria. Again, 30 families. Notice again, there is no presence of Arab world. We were not presented in any of these international genomes projects for a very long time. Another project came, it also called 1000 Genome. Now they did more. The first one was 12. We did 27. Now we did 1000. Again, technology got cheaper, and we'll show you in the next few slides. So they did whole genome sequencing here, a complete destructive in more than 1,000 individuals from multiple world population. Arab world was not part of it. And now this is not very up to date. There is many nations have launched their national genomic projects and dedicated significant budget to this effort. Why any nation will do that? It's important to know the makeup of your population. We said we're similar in 99.6%, but we are different. And this is different even between different ethnic groups in the human species. And why we want to know why we're different, because this can explain different diseases, carrier rate for diseases, etc. All developed therapies, all of this important. Plus the technology became cheaper, so now we can do it. The human genome, I forgot to say, it costs around $3 billion to do. Now it's we'll see how much this cost dropped. So one of the most famous national genomic project, I think it's one of the leading worldwide, is the Genomic England. They started with 10,000 into the 100,000, and then completed 500,000, and now they're going to 5 million. It's one of the most comprehensive and detailed, and it's open source for all the researchers to look and do whatever studies they want to do, comparative study, whatever studies they would do. Many in the US, actually, it's not only one, but this is the most biggest one. It's called All of Us Project, biobank/sequencing. In the Arab world, we have Saudi genome, we have Qatar genome. There is, sorry, it's not updated. We have also, and we heard about the Egypt genome project. There is some small project in Tunisia and Morocco. We worked with them in some recent project related to COVID. So there's a few attempts in the Arab world. It's not really to the level you see it more in the Western populations. China, of course, has its own project. They call it Precision Medicine Initiative, and they even developed their own sequencing technology from scratch. So let's go back and talk about the DNA. Where I can get DNA from? You can get DNA from any fresh tissue. If you take a skin biopsy or liver biopsy or from any organ, you can get DNA from blood, because we can get DNA from lymphocyte, from cytology, and from a urine sample, akramakumullah, from cytology in thyroid nodules, from anything, you can DNA. From biobank, they usually keep the tissue frozen to -80 or -20, so we can get DNA extracted easily. Actually, the samples usually good for both DNA and RNA, the fresh and frozen. From fixed tissue, in many hospitals, we use fixed tissue, especially in the pathology department, and formalin. Formalin usually break the bond and make the DNA degraded, but we still can get DNA and even RNA. Not the greatest like the fresh one. Forensic world, this is a whole story. They can basically get DNA from anything. I mean, I worked for some time in the New York Medical Examiner Officer, and they have one of the largest genomic forensic lab next to it. From anything you can imagine, they can get DNA. Even if you touch this glass, they can get DNA after you. It's really scary, but this is actually very good for their criminal investigation. So don't even compare what we can do in clinical versus what they can do in forensic. And this is Dr. Bambu. Again, I like this picture when you're looking at the Neanderthal's skull. That's what he has to work with. Samples 40,000 years old. يعني مدفونة في الله يعلم how it was the condition. They found it this labor and random digging. So it has a lot of microbe. All this brown strain is actually microbes, some contamination. It has some of our modern human DNA, and it has this little blue DNA, what he is interested in. So you can see how hard it was got to clean it up to convince the scientific community that actually he's sequencing this specific species. It's not a bacteria or microbe somewhere. So it wasn't a very easy job. So he extracted the DNA, mitochondrial DNA, which usually this specific DNA came from the mothers. And he was able to extract it, to clean it up, to make library, to sequencing. And not only this, to do the entire sequencing, like what we did for the Modern Human Genome Project. So this can tell you how back you can get DNA, 40,000, 50,000-year-old samples. And then don't think only it's a human, especially for those who don't know. I mean, DNA present in any living creatures, including shells, flowers, corns, anything, you name it. There is DNA in everything. And there is actually genome project for many of these creatures, for horses, for dogs, for different fruits and vegetables and bacteria, viruses, etc. So we said we are different. What makes us different? The different could be only change in one letter. We call it single nucleotide variant, SNP sometime, you will hear it. Or it can be group of change of bases. So it could be this segment of biochemical bases. When it's large enough, we call it multi-nucleotide variants. Could be small base pair, either insertion or deletion. We call them indel. Or large, it can take actually segment of the chromosome. So we call them copy number variation, either loss or gain. Or they can get inverted. يعني what here got here, on here got there. This is very simplified way. So we call them inversion, and they can translocate it. What in the chromosome 1 can go to chromosome 15, and the vice versa, etc. It's called translocation. Or we can get aneuploidy. We can get the whole new chromosome. For most famously, Down syndrome, trisomies, they can have 23 chromosomes. So this is what makes us different. Different doesn't always mean we make us sick. It actually, majority of the time, this changing in SNP on chromosome, they're not actually, they're benign. It doesn't cause anything. They can account for some trait, for example, eye color, hair color, skin color, different things that not related to health, to disease. And some of these changes can cause diseases, especially if they're genetic diseases. We call them Mendelian diseases. We know over 6,000 Mendelian diseases. So there is a lot of discoveries with improving technology. And some of them can be involved in very complex diseases or trait, like heart diseases, Alzheimer, all of this. This is very complex. You cannot just say this gene causes disease. It's more than that. And they also can, some of the changing can make us susceptible to certain disease. Some of them cannot be as actually protective. Famously, some changes in the African population that gives them some protection against malaria, some sort of type of malaria. Same, there's some discovery, some protection against some type of HIV infection. Same with COVID. We discovered some traits that it actually can cause the people get more severe disease, including young people. So this is what CNV and SNPs usually does. How we look at this is different things. There is too many technology. We talked about PCR, so take a small piece, make multiple copies, and then use it for another project. I mean, COVID PCR, for example, we test for a specific part of the viral genome to see if we're infected or not. Some of the changes you can see it by microscope using technology like FISH, fluorescent in situ hybridization. You can see it here, and this, we actually do some sort of hybridization with certain fluorescent stain. And you can literally see how many copies. You should usually have 2 copies from each specific area of interest. But sometimes they get fused together, translocation. And sometimes you can get multiple copies, amplification, copy number changes. MLP is the same way, it will not be used really now. Array CGS SNP array, again the same. Array CGH, basically we make a chip, we label a different part of the genome, 400,000 area, 250,000 area. An array 6 million area, we ship it and we look for changes, what gains and losses happen, and it's is it located in a gene specific disease. Sanger, we talked about Sanger. You look at everything, although it's a short read, but you look at everything. You can of course do Sanger on very in a specific segment of the DNA. You don't have to do Sanger on everything. It's actually very expensive and nobody do it this year. I think they stopped since 2003 to do it on the whole genome. And then NGS, everybody will say NGS, next generation sequencing. We'll give it more attention in the next few slides. So what is genetic mutation? Again, it's a change. All of us have genetic mutation, all of us, more than one, or we are carriers of some of them. We inherited some of them from our parents, and many of them occur randomly or just we don't understand, and different environmental factors, especially smoking, diet, etc., can lead to develop more mutations. So we're a walking body with more than 200, 1,000 mutations in our body. Not, alhamdulillah, يعني لطف الله فينا, إنه these not all of them turn to be a diseases. So this is really something science can never explain. Now, we can relate that genetics or some genetic changes related to the most common diseases that affect us. This is a top 10 disease killer in the US, according to CDC. And they're not really different from any part of the world. Most of it really depends, especially now in our part, yani, you can relate diabetes and cancer and heart disease is still the number 1, 2, 3 in because of the obesity lifestyle. So they are similar, really, it depends just on the order where you are. All of them, you can pinpoint to some sort of genetic causes. طبعا يعني with exception of accident, يعني car accident happened because maybe sometime maybe you have some condition or got something that led to an accident, but everything else, يعني especially when you look at cancer. Cancer is a genetic disease, and we'll talk about it. And the disease of each cancer is different. Each cancer is caused by a driver mutation, either you inherited or developed it. Some disease is complicated, like cerebrovascular disease or heart disease. It's really hard for most of them to pinpoint to one genes. Alzheimer is the same. I mean, we know some genes, but it's not the only one. We can really say this is what caused it. But susceptibility to some infection, we can pinpoint to some of the genes. I explained to COVID-19, the most recent one. Many genes discovered, at least 27 gene variants discovered that related to severe phenotype or severe infection of COVID. So in summary, this human genome project grew up different methods for technologies, next generation sequencing, different alignment technology, different method to do it. So it wasn't only, it put the order of the book and also helped to open the door to develop more technology and more way to do the analysis. This is me with Dr. Watson. My Facebook gave me a note yesterday. It's been 10 years since I met him in his office. He's still alive, I think. He has some interesting personality. You can read about it and opinions about different things. So anyway, in 2006, remember the first cost was $3 billion. Took them 13 years to do it. 2006, just 3 years after the last draft they published, it now become $22 million. 1 year after, it's become $1 million. Watson actually sequenced his own genome, and it took only 2 months. It took them 13 years in the beginning. 2008, Sanger Institute in UK sequenced 300 genome only in 6 months. 2008, cost dropped again, 10,000 to 4,000. This is for research cost. Clinically, we started to do a whole exome around 2011, 12, and then whole genome become really trend in the last 2 years. This is the cost for clinical exome today, $800 to $1,500, whole genome, $3,000, $7,000. Research can go much cheaper than that. And it can be done in hours. 2-3 weeks ago, I believe, there was a huge genomic forum by Illumina. Illumina is the major provider on the sequencing technology worldwide. You can say 90% of all sequencing in the world happened on Illumina platform. This is one of the newest next generation sequencing technology. Their CEO announced that they have this new machines. Although it's like he made it like in a style of Steve Jobs announcing iPhone. This new machine can do 20,000 whole genome each year, which it costs you only $200 to do it. So it's going really low, but you need to have to scale, and you need to have, of course, the money to make this functional. Another Illumina machines able to generate more than 6 terabytes of data in just 48 hours. This is a lot of data. So the question comes when you think about all of this, what really this genome did to me, and what information related to human health came out of it, and what do we mean by personalized medicine or precision medicine, الطب الشخصي أو الطب الموجه. Precision medicine is really the balance between have a cost control, so you can implement this to treat people or to help people, and also to balance between this scientific explosion of data. There is tons of data get produced every day. We cannot keep up. We cannot keep up with analyzing them, making sense. In fact, we still don't know about the function of many genes. I think the latest I read, we know the function maybe of around 1,600, 2,000 genes. Many of them we still don't know can correlate it to specific function. So this is a balance of precision medicine when we try to get things into the clinic. We'll give some example. This is the next generation sequencing. This happened to be Illumina. Again, I told you it's not really commercializing any ad for Illumina, but this is what you will find. At least if you go to Zen centers who do sequencing, 9 of them will have Illumina. It can do many things, direct DNA, RNA, epigenetics, methylation, chip, many things you can imagine, single cell, etc. So a lot of data, a lot of method, and different applications, and the cost, the drop of cost, what made them kind of popular worldwide. So for us, we mainly, at least in the clinical work, we focus on these 3 areas, doing exome or only the coding area, because it's easier to do, to interpret and to do, and it's still a price cost, it's doable in the clinical setting. Genome is more advanced, you know, do everything, the cost will go higher. And still we do targeted genes panels. We select 50 gene, for example, for patient with mental disability, 50 gene patient with certain cancer, etc. Because again, we don't want to manage the cost, the turnaround time, and how we can help our patient in a real time. This is, of course, different than research. Research, the prior slide, you can do it in anything. Different techniques and methods. I'm not going to go in details, but it's not only one chemistry. I wanted you to, if you are new to this, there is tons of techniques. Illumina pioneered one or a certain chemistry. Ion torrent thermofisher pioneered another one. So different companies came up with different things, and different scientists come up with different techniques. You really do select what is work better for you. So this is, if you want to summarize the next generation technology, it wasn't really next generation. This is the first generation. Sanger is the main one. We use it in a human genome. It's been used since it was discovered in the 70s until early 2000. It used a certain way of chemistry and fluorescence to visualize the space pair using electrophoresis. It do a short read, relatively medium size, 500 to 1,000, and very labor intensive and expensive to do if you want to do the entire genome. And then we come to next generation sequencing, Illumina, Ion torrent, etc. Roche has their own platform in the early day. Now it's high throughput. We can do it quickly, different chemistry, some of the different reaction. However, they only do short read. Now, what do you mean by short read? When you do sequencing, you really can imagine you have a piece of paper and you cut it to very small pieces, fragment. And then you try to rebuild, make many copies of them. We call it coverage, and then try to put it together. And put this together, referencing again, it's in a field human genome reference down. So different copy up. After you cut it, you want to rebuild it again, alignment, and then through it. So when you have very short copies, sometimes you lose things. So that's what made it very difficult to complete the draft. Now, the third generation technology backed by Oxford Nanopore, they were able to develop technology and chemistry for what we call long read. You can get to up 100,000, even 1 million base pair fragment length. But you don't really have to cut this piece of genome in 2 million, very small fragment. You can actually make it longer fragments. So when you reorganize it, you don't lose anything. And this is what made it possible for a project called telomere to telomere, which we talked about in the beginning. They use this technology heavily. They included Illumina, but they also use this technology. So they were able to fill the gaps in the genomes. Bioinformatics, you cannot talk genetics without talking bioinformatics or computational biology. This whole field exploded after the Human Genome Projects. We call them the dry lab scientists. They don't go and do experiment in the lab. They just sit by their computer and their databases and do the alignment, everything we need. So after the machine gets the output, it's called fast Q, gene, the quality control, and you do the read mapping, trying to remove any noise, duplicate, etc. Then you refer to variant calling or different genome, and then you can read and identify any potential variant or changes in the genome. It's a whole world by its own. It's very difficult and it's extremely to do it. But now there is multiple software and pipelines and even free courses online, at least to know the basic of it. And to work with, there is also open sources. You can work yourself trying to find what the mutation or what is the variation. And then the question is how I can use it in the clinical care. You can use it to diagnose patient, to trying to find some risk score if you want to prevent some disease, or you can use it to detect some novel organism like what we did in coronavirus and Ebola virus, and all of this virus has been discovered every day, Zika virus. All of these things can be detected by next generation sequencing and other technology. not only human genome. So there is, as we said, there is large-scale genome project nationwide. There is microbiome project, different viruses, bacteria, nematode, etc. There's comparative genome projects, big scale one, like the famously banned genome project, telomere to telomere project, all of these. The project related only to RNA-seq and methylation and epigenetics. And there is many, many, many others to do. There is consortium disease-based. We'll talk about the tumor cancer genome atlas. There is consortium, for example, some psychotic disorder, etc. So it's getting a very, a lot of detail, a lot of subspecialty, a lot of focus. And also it raised some challenges because you really need to educate the provider who somehow either now or in the near future, they will have to deal with this data. It's not only research, it would be part of their patient care. And we still, unfortunately, at least in many medical schools, we don't really give enough attention to the student or even in the training, to the people to learn really deep what they will get to face in the near future. Issue of ethics, it's always been there, but we are much better shape now. There is MITI guideline and ethical agreement in the community. Education public, this is one of the efforts we do today. And also there is many, many in Arabic on different languages, educational materials. How are we going to store this data? You know, generating data could be relatively easy, but storing them and making sense of them, interpreting them, this is a very difficult question. And who will store it? And should we store this data for how long? All these questions came with a challenging of the availability of this technology. Sometimes you find things you don't want to find. Sometimes we do sequencing to discover, to find answer or discover what causes specific disorder in this patient, where you find some incidental finding. We find, مثلا, patient has Huntington disease, some like terminal disease. We're not looking for it really. So thanks God, American College of Medical Genetics, European College of Medical Genetics, and the others, like they put lists every year to update what you should report, even if it's incidental findings and what not. Also gives the freedom for the patient sometimes to make their own decision. So it's not perfect, but it's way much better than when it started back in the beginning of the century. But this is what we wanted to talk when it comes to ethics. We're not really making genomics to create what we call designer baby. Many of you heard about this big ethical question of this Chinese scientist a few years ago when he used CRISPR technology to modify the baby to make them not having HIV. This wasn't intended to make us select our baby. Francis Collins, again, he said, we're getting closer to perfect kids by caring families, by good education, good food. Then what we will get ever from genetics. We're not going to use genetic to put the smart gene in your kids or the blue eye gene in your kid. We wanted to use it to prevent potential disease or to treat potential disease. Traits mainly controlled by environment, and especially when it comes to intelligence and all of these things, and sports, excels, and all of these things. So I will go quickly about some genomic clinical applications. So sequencing usually, at least heavily being done, it's ordered for many, many patients in the medical genetics clinics, especially for looking for rare diseases or unrecognizable condition. We do exome or genome. This is known. It's most of the countries, they do it because of the cost. We also do it for, so as I said, mainly for Mendelian genetic disorder. We do it also for macrogenomic, which means look for, to see if this specific variant will make the patient, will make certain drugs work for this patient or not. So this is what we call it for macrogenomics, the right medication based on genetics. We use it a lot in cancer because there are a lot of personalized cancer treatment, different type of cancer. We also use it in infectious diseases and screening, diagnosis, etc. So we use it a lot, or even to develop vaccine. Cancer is challenging because of the disease of genome. Each tumor is different and each cancer patient is different. You either inherit cancer gene, which is very rare, alhamdulillah, or less than 10 to 15% of the cases you inherit cancer gene. Most of the cancer gene is actually caused by driver. There is something that happen in the genome lead to certain gene to drive cancer. We call them driver. Because of this driver, we were able to identify many new targeted therapy, which means you cannot give this therapy without the specific mutations. And this is very important. And this is where the term personalized or precision medicine came. It started by glioblastoma, one of the deadly brain tumor, because this influential politician got it in 2008, Edward Kennedy. But however, it led to develop one of the biggest consortium for cancer. It's called tumor cancer genome. It has many, many different kinds of cancer being sequenced using different sequencing technology today. BabySeq, there is project actually that sequence babies the moment they get born. And this is still have some ethical questions. For how long we should keep it? Do we share it with a baby? All of these things. And it helped many babies also to treat some of the, vital condition early on. Maternal blood, maybe many of you know, we can obtain blood from the mom in early pregnancy, and we can know if the baby, for example, has trisomy or not. TDC, or direct to consumer, it's very getting public, especially in the US and the Western community. And this is example of one famous company, 23andMe. Google invested in it. So this is one of their pharmacogenomic reports. For example, if you have what different certain genotype, how you respond to interferon therapy of hepatitis C. And they report at least for 10, 12 condition, pharmacogenomic plus many other things. In progress, complex trait. This is the most difficult one. How you can explain the interaction of environment, different genes, not only one, to develop risk score or even therapy for certain condition. This is still difficult area, and I think it will take us some time to try to understand let me say 20% of it. We're still in 20% of the Mendelian disorders. There's still a lot to do when it comes to complex disorder. This paper kind of summarized it all, and I think I would end with it. This is where we started, genome. We put the letters in the book. We know where it is. Then we started to understand the biology of function of many of these. Then we started to use it clinically in exomes on targeted gene panel to diagnose and treat some of the condition. And now we're going beyond. We're trying to improve the effectiveness of healthcare and also advanced medicine and understand more the complex interaction. So genomic medicine is just around the corner. If it's not in the corner already for some subspecialty like cancer and genetic diseases, there is a lot of now space for personal genomics, so direct to consumer, but be careful that everything that advertised is true, especially when it comes to things like nutrition, well-being, skincare, and genetics. This is not really backed by a solid science. So don't just order anything just because of the fun. Sometimes these things need to be ordered by the provider to better and help you in understanding them. Genomic medicine is just the beginning of big data. And the question is how to work with this data, how to use different tools, bioinformatics, computational biology, AI tools to make sense of it and to implement what is really called personalized healthcare. With that, I will end. I'm sorry if I took some time. I will try to answer some question. I think we have 7 minutes. So Summer Ashraf asked, CRISPR. Yes, CRISPR, there is different editing tool that's not very famous, depend what lens you want to take of it. But when it comes to its application, CRISPR get most famous because maybe it was one of the beginning and one of the actually, there is a lot of companies and some clinical trials that have been published, for example, treating thalassemia and others that have been used, but CRISPR is the most famous one. In my personal opinion, it still needs time to prove itself at a wide scale, I mean, in the clinical use, because I'm not aware of any application. There is some CRISPR diagnostic testing that's been developed during COVID, for example, and some other in the way, but not really a lot being used. Nanopore, Oxford Nanopore, Ali Al-Anani. Nanopore, as I said, it's a third generation. It's been used. It's still a research tool. We do not use it in the clinical. I just used it this year, Nanopore and BackBio, in a very interesting project because of the long read capability. So it is still a research tool until now. It's not in the clinical practice yet. I think that's all. And again, مرة تانية, please forgive me because I really don't, هذا خطأ مني. I cannot really translate every word in Arabic, but just to give you a flavor on what's happening. And there is still a lot of error rate. There is error rate. None of these technologies are perfect. None. That's why this project I worked on, and specifically we used 3 different technologies. And we used Illumina Nova 6, we used Nanopore, and we used BackBio, because each of them has its own limitation and error. So we use different one, trying to come up with a draft, trying to reduce the number of error and gap. None of them are perfect yet. That's why we're giving this lecture, because there is a lot of room for you, especially the younger you, who want to consider this career. There is a lot of things you can do when it comes to working with technology, developing technology, improving technologies, improving pipelines, artificial intelligence, bioinformatics, doing functional work, functional biology work to understand what is related to us, coming up with diagnostic tools, or even or examining some therapies. So yeah, so that's one of the, we just want to give you, يعني بنقول كده فاتح شهية لكم, appetizer for you, so you can think more about it. It's very wide open. It's very wide open. Sometimes the news take a big title, like make you feel that خلاص انتهى, يعني ما في شيء تاني you can discover. There is actually a lot of things to work on. Summer, yeah, there is other genetic editing tool. You can email me or send to me, and I will send you a link. But they're not as famous as CRISPR. There is other things that people are working on it. It's not my area of specialty, but I mean, I work with somebody who works in editing tools. They're not famous as CRISPR, but there is other things that you can do. There is something by base pair, something like that. طيب, I think it's about time. طبعا let's end this بالعربي. الجينوم زي ما قلنا بالعربي، الجينوم كافة المادة الوراثية الموجودة داخل أي كائن حي. الجينوم بالعربي، الجينوم البشري اسمه المجين. الجينوم علم لا يتعلق فقط بالإنسان، يتعلق بكل الكائنات الحية. الإنسان يكون من أكثر الكائنات اللي تم دراسة الجينوم حقه، يعني لسبب واضح، سواء دراسة تاريخية بالأنواع القديمة من الإنسان، أو دراسات الإنسان الحديث. تقنيات مختلفة درست، لكن كذلك درست أنواع مختلفة من الكائنات الحية، بكتيريا، فيروسات، بعض الدود، الفواكه، الخضروات، الخيول، الجمال، التمور، النخيل. يعني ما أعرف قديش أقدر أقول لكم، أشياء كثيرة درست وليها جينوم بروجكت. نرجع للكائن الحي الإنسان، كثيرة مشاريع جينوم فرعية صارت لأمراض مختلفة، لصفات مختلفة، استخدام تقنيات مختلفة. في الوقت الحالي أصبح كثير خط، لا نستخدم فقط الDNA لمشاريع الجينوم، نستخدم الRNA، نستخدم الإبيجينيتكس أو فوق الجينوم، نستخدم حتى دراسة single cell جينوم يسموها، دراسة الجينوم حق خلية واحدة. شوفوا لأي درجة وصلنا، يعني إحنا كنا ناخذ مجموعة خلايا ونخرج منها المادة الوراثية وندرسها بالإجماع، ألوف الخلايا، مئات الخلايا. الحين نقدر ندرس واحدة خلية، واحد جينوم للخلية الواحدة، نقدر نحدد بالضبط فينه. بل بعض التقنيات الحديثة تقدر تشوف على السلايد، اسمها سبيشال ترانسكريبتوم، فين بالضبط الخلية اللي بتدرسها، وش الترانسكريبتوم أو الRNA سيك تبعها. شكرا جزيلا، وإن شاء الله نراكم في محاضرة قريبة. وأشكر أسبوع العلوم العربي على الفرصة، والسلام عليكم ورحمة الله.